So we'll continue with our discussion on EEG. Well, we only spent 5 minutes in the last class, so I'll just recap what we had done. So it's a technique in which electrodes are placed on the scalp, um, on the surface of the head, and it's a very popular technique, almost 100 years old now. The first recording was done in as early as late 1920s by Hans Berger. So you have a metal electrode and you put some sort of a gel. Usually we, back in the days when I was an undergrad, we used to put salt water, which also works pretty well. uh, is to increase the conductivity between the electrode and skin. It's a very popular method. It can be used on infants, on people who are meditating, doing different cognitive tasks, and so on. So a bunch of electrodes stuck on the on the head. Um, so usually we have certain conventions which dictate how these electrodes should be put. This is, for example, a very famous or popular 10-20 system. Um, the electrode positions are determined by the names depend on which lobe these electrodes are placed on. So the electrodes here are called occipital electrodes. So O1 and O2. One side has even numbers. The right side is even. The left side is odd. And then temporal is this area corresponding to the temporal lobe here. This is parietal. Then this is central, and this is frontal. This is frontal pole. Um, and then this Z is the center line, the midline here. So it's either neither this side nor that side. So this is FZ, CZ, and PZ. This is the 10-20 system. You can read about how it's done here. Won't get into that much detail. Nowadays, you have denser electrode arrays. So the system that we have in our lab has 64 channels. In fact, we can go up to 128 even. But the data that we have collected and the data that you will have access to have 64 channels. So now you have not just O's and P's, but also PO's. So it's parieto-occipital. These are centroparietal and so on. Fronto-central, FC3 and so on. So now because you have more electrodes, the names are defined in a similar way. And then usually there's a ground and a reference electrode. I will tell you about these in some time when we talk about referencing. Okay. Yeah, and nowadays the latest ones have even more electrodes, very dense electrodes. For example, this company called EGI has this very dense neck, um, which has 256 channels. So it's even on the face, as you can see here. Um, so that's that. Now the math that we are going to discuss mainly in the next few classes, as far as EEG is concerned, is going to be around this problem of what are called forward and inverse problems, which essentially mean that if I give you the voltage distribution, the voltage on all these 64, well, in this case 128 channels or 256 channels, can you tell me where the source is in the brain that is producing this voltage distribution? This is called an inverse problem because from the voltages you are getting down to the sources. And the forward problem, which is a more straightforward problem, is that if I give you a source in the brain, the source we will see is often modeled as a dipole. Given the dipole, can you tell me what the voltage distribution is going to be on the scalp? Okay, that's the forward problem. So we will be discussing the biophysics of EEG mainly in terms of forward and inverse problems, and that's going to take the majority of our time. And then very briefly, we will talk about connectivity and causality measures, which are very popular in the field. And that's that. Um, that's our agenda for this module. Okay, so the references that I'm going to follow are mainly these ones, Hales and Gretsch. Very nice reviews, but hard to understand. So you'll have to spend some time reading this. This uses a fair bit of uh, um, linear algebra. So you'll have to brush up your linear algebra to do this. Um, and then there are a couple of lectures here that are also very useful by Hoffe and Nolte, which you can actually even download as PDFs. Okay, so let's first talk about the EEG data. So you can get this data from the course web page. It's the data that we collected when a subject, in this case now a human, was staring at a screen while we collected data from 64 channels. The subject's head was stabilized through this EEG, this chin rest here. Subject's eyes were monitored using an eye tracker that you see here. Change my and the subject was performing a fixation task, which means they was just looking at a dot on the screen while we presented stimuli at 8 different orientations, all full screen. So the gratings were presented for 800 milliseconds with an stimulus duration of 700 milliseconds. And we showed 8 orientations, but the data that you have is only from one orientation. Okay, so now let's look at the data. We'll look at it in two ways. First is using a customized program that we have written, and the second one is this more standardized field trip package that we are going to look at in MATLAB. So this is the customized program. So we can ask when I show a stimulus, where is a response coming from? So now you have to look at the data from 64 electrodes at the same time. So that's what is shown here. To get to a plot like this, you have to sort of have an idea of where each electrode location is, and that's an important factor. Usually the reason why we have caps like these is because then you can very easily know where the electrode position is. They can't move too much. However, you don't necessarily need to have a cap. You could, if you wanted, stick electrodes one by one at these locations if you knew where these locations were. Okay, and then 1020 system, that's the idea that you can sort of measure with a tape what the approximate locations are and then just part the hair there and put some gel and stick the electrode and put some tape. And that's what traditionally people have done. Nowadays there are caps, so you just put a cap like so and it will have some holders to place the electrodes and then you have a good idea where the electrode locations are. All right, so the evoked response, which we are going to look at first, is just the average of the LFP data locked to the onset of the stimulus. And what you see in this data is that here, these are the electrodes that are showing some response. See that? This is a response. However, it's not localized to one location. There are quite a few electrodes that are showing a response. And maybe you can also see this slightly dense packing here. This actually corresponds to the gamma rhythm. So the line becomes slightly fatter after the stimulus is presented. Okay, and this happens to be the occipital area. This is where the visual processing takes place. So you can see that the sensory stimulus, the visual stimulus, is mainly producing a response in the occipital area. And not only that, it's a lateralized response. Often, even though the full screen stimulus is, going to activate both the hemifields, often the responses are stronger on one side as compared to others, to the other side. We don't know why that is the case, but this is something that we have seen in many subjects. So now you can ask, okay, I want to look at the gamma response. So let me look at the gamma response. So I'll take the Fourier transform of all the 64 channels during the stimulus period and the baseline period. And then this is how it looks like. Okay, so FFT, the stimulus duration is now 500 milliseconds. Baseline is also 500 milliseconds before the stimulus onset. And this is in green, this is in black. And here, notice what is happening. There is this peak here at very low frequencies. This is the alpha rhythm. Okay, and the alpha is stronger in this on this side. It's also there here, but it's not only localized to the occipital areas, it's all over the place. You will see some alpha even here. It weakens in the frontal areas, but it's there here. In addition, there's a second bump. This is the gamma rhythm. See these bumps here? These are the gamma rhythms that you see that are more localized in the occipital areas and in this subject on the left hand side. Okay, so instead of the raw spectra, if I look at the change in spectra, how much did the power change from baseline? You can now see the gamma rhythm, this peak here at around 40 hertz, and then there is some alpha suppression as well. Okay, this is suppression in alpha, this is increase in gamma. So the stimulus, when the subject was looking at the stimulus, we see an increase in gamma and a suppression in alpha. And it's mainly localized to the occipital areas, but it's also sort of spreads to the parietal areas here. It's lateralized on the left hand side. And in the frontal areas, you don't see much activity, much sensory evoked activity. Okay. Yeah, and if I zoom to this particular electrode and zoom it in and show you the raw spectra, this is how it looks like. So this is the alpha rhythm, this is the gamma rhythm, and there's alpha suppression and gamma increase when the stimulus is presented. This is for electrode 61. So this brings us to a few questions that I'm going to now ask, and this is what we are going to spend a lot of time on. So I see that there is gamma being generated, which is leading to this peak here, but where is it generated? You can see that gamma is generated, is recorded at quite a lot of electrodes. A lot of electrodes are picking up the gamma activity, all these electrodes. But is it the case then that this entire brain area here is generating gamma? Or could it be the case that the source is very localized, but it's sort of the activity spreads out, and therefore, no matter where you have an electrode, as long as it's more or less close to the source, you can pick it up. So this is this classical question of source localization, which we are going to discuss, and which will then give us an answer to where this gamma is generated in the brain. Another important question is that where is the, does it matter where I put my reference electrode? Remember, in these settings, there's a reference electrode. This is the reference electrode. Now, if I use a different reference, will that change the response? That's another question that we are going to address. We are going to briefly talk about current source density, and then mostly we'll talk about source localization. So these are what we are going to discuss today. I mean, in the next few classes. Okay. So the analysis that we are going to do is all based on a particular software called Field Trip. So this is a very popular software in the field. This and EEG Lab are sort of two, softwares that are used very often. So let me show you how this works by running the commands on Field Trip. So you'll have to download Field Trip and okay, so on desktop, yeah. So this is EEG data resources, and I'm somewhere else now, so I'll go to. So on the course web page, if you go to if you go to the course web page, you will be able to download this EEG data resources, CNS. Here's the course web page. EEG data. So if you click on this, you'll get to EEG data resources, and this is that folder. And in this folder, you have the data here, EEG data all.mat. And you also have electrode.mat, which tells you the position of each electrode. And then we have got a few other programs that we'll be looking at together. So let's look at display data.m. So all you have to do is edit display data, and then you get to this program. So if I size of data. Okay, here's the data. Data.trial. So notice the layout here. This data file, data.trial, has 188 entries. So what is 188? It's the total number of trials that we have. So I've got 188 trials, and each trial corresponds to a matrix of 64 crossed 438, 64 electrodes, and 438 data points. Okay, and this time tells you what those data points are. They're all the same. So dot time, first one. Yeah, so the time here is from minus 0.7 to about 1. Okay, 438 data points. The sampling rate here is only 250 hertz. So that is why there are fewer points. Now, this is what multi-taper will do. Let me first say plot the evoked response. So when I run this, there. This is similar to what we had seen earlier. You can see that the evoked response is stronger here. But a field trip allows us to do some very interesting things. So for example, I can choose these electrodes and click here, and it will give me the average evoked response for these electrodes. So you can see that there's this nice dip here and a peak there. This is the evoked response. Okay. And let's look at the time frequency response. So I choose the window length and set some other parameters here which you can read, and I can now get this time frequency data and then plot this. So let me just run this. Okay. Um, can you guys see the screen? Is it are the plots visible enough?
Can you please zoom it a bit?
Okay, so so this is as much as I can zoom, but I will show you why this multi-plot thing is so useful. So this command has generated 64 time frequency difference spectra, and you can see maybe some red here and blue here, and these things are just all blue. So I could actually, if I wanted, say, tell me just this particular electrode and click on this, and then you can see it much better. See that? So this is 0. There's the slow gamma, and there's a fast gamma here as well. And this is the alpha suppression. This is for a single electrode, PO3. I could actually choose all these electrodes that were that produce a good gamma response, and this is the mean of all of these. I'm hoping this is more visible. You guys can see this, right?
Yes.
So this is the time frequency spectrum as before, and the stimulus onset produced a big gamma response. This is very similar to what we had seen earlier using that more customized code. But I could even do more interesting things. Let's say I choose this frequency like so, and say, okay, between 200, well, okay, let me choose the range between 250 and 750, which I had done here. So 250 to 750, and now show me what is called a topo plot, which is how much gamma between this time period and this frequency, how much gamma power is there in different electrodes. So now when I click on this, it produces this what is called topo plot. Okay, and this is what tells you that the gamma is really localized, at least on the scalp distribution on the left hand side. Okay, this is pretty neat. It's a, does something happen when I click on this? It gives me back. This is again the time frequency spectrum for all the electrodes that are between this rectangle. So you can play around with this code and actually learn very quickly about how the voltage is distributed on the scalp and average of different electrodes and so on. Okay. So EEG analysis, a lot of it is actually just trying to visualize. Visualization is tricky because you have data from so many electrodes. And that's another reason why these software packages are so popular in EEG analysis, because a lot of it is just good way of displaying the data. Okay, the math is not hard, but how to display and how to customize and how to choose time and frequency ranges and so on, that's what is more important. So if I choose these ones, you would see this more strong alpha suppression, and then there is gamma here as well, but it's not as strong as the gamma on the other side. So in fact, now here if I do a color bar, will it show me a color bar? Huh. So here it's from -2 to 3.5, and if I do this side and do a color bar here, this is this is 12. So that is why this is barely visible on this side, but it's there. It's just weaker gamma. Okay, so this actually is a very good data set. This is one of our subjects who had the strongest gamma response. Um, and usually in subjects in EEG recordings, we don't get very strong gamma. It's weaker than what we get in with invasive electrodes, but this is a very good, particularly good data set. So you have you can see whatever you know important things are there in terms of alpha and gamma oscillations in this data set. Okay, any questions on this? This is quite straightforward, right? We have gone through these kind of analyses before. So now we will get to the question of referencing. Okay, so tell me, here's the electrode to which all other electrodes are referenced. Remember, whenever we talk about potential or voltage, we talk about voltage. It's essentially voltage difference between two electrodes. Okay, so then, every electrode, I put one electrode here, and then when I look at the potential, it's potential difference between this electrode and some other electrode, and that some other electrode is the reference electrode. So all the signals are referenced to this one electrode, which is at the center here. But there's another electrode here called the ground. So does anyone know what's the difference between the reference electrode and the ground? And why do you have two electrodes? You don't know, try to guess.
Grounding is used to get the zero value. Means when we have this
What do you
ground,
mean zero values?
basically, you also have the noise. No? Grounding is always done to reduce the noise as well, because you have like one positive and the other negative. If you are just working with positive and assuming the other as zero.
No. Well, your first part of the answer is right. I don't understand the second part. Explain it one again.
So basically, to form a complete cell, you have one positive and the other as negative. Okay. Now
Yeah.
this negative is taken, or if when you have this reference electrode or basically voltage also, you have to have two terminals, and one is then taken to be grounded, and this is taken to be zero. Otherwise, if you just leave it open, it will catch noise, and then your referencing and everything would be spoiled.
Well, so referencing is not spoiled per se, because you
Yeah,
only need two electrodes to have this differential recording. So let me explain why you need the ground. So the second part of your answer is correct. Where shall I? Okay, I will just draw it here. So pen. Now, in general, so suppose you've got one one electrode, which is your, let's say, O1. This is a proper electrode, and then there is one reference electrode. And usually they are sent to some sort of a device. Uh, in this case, what I'm drawing is what is called an operational amplifier. And there is a way that the output of this is O1 minus ref. Okay, so you are looking at the voltage here with respect to the voltage at some other location. Okay, so this much is actually fine. You can live with just this much, because all you want really is how much activity is in this part of the brain where O1 is sitting as compared to the reference. But usually when we put electrodes on the scalp, so now we have got a person here, very happy, and we have got lots of electrodes. If you see an actual recording, you will see these things in action. If this person simply touches, um, you know, an electrode or some metal object or something, you will see huge noise signals in all of these electrodes. Okay, in fact, don't really need to, for the person to sit, touch anything. Even if there is no grounding, you will see a huge oscillating thing. This is not gamma, this is line noise, 50 hertz. Now, why is that? The reason is that our bodies are like antennas. We actually pick up a lot of signal from our ambience, and from our environment. What kind of signals are these? These are all electromagnetic signals. Tube lights, fans, motors. In our EEG lab, there's a this bunch of computers that are running. Um, there are there's a, what is this thing called, a ducting system that is running. So all of these run on 50 hertz, and they essentially generate a lot of noise, and our bodies simply picks it up. So you will see this huge noise just because from the ambience there is, this is like, from the ambience, this is like a tube light or a bulb. You're seeing a lot of electromagnetic radiation, and our body is just picking it up. So it's essentially some charge developing on the surface, which is getting picked by the electrode. So a good strategy is to have one electrode which is connected to the earth, the ground, and it basically just takes all this charge that is developing and shunts it to the ground. Not only this, our device that we have, which a device like this, which is this in this case is going to be a preamp, for example, preamplifier, that also picks up this and electromagnetic radiation. So we also connect this to the ground. What do we mean by ground? So often these are dedicated earths. So we actually, dig a big hole in the ground and put a lot of metal and charcoal and water and salt in that, and have a big copper wire, which then, provides as a like a dedicated earth to which we connect this electrode system to, and this thing. So only the most sensitive equipment is then connected to that. It allows a low impedance, a low resistance path for all this induced charge to get to the ground. So it simplifies our recording. Once you ground properly, you will see that this is much reduced, much smaller than before. Okay, so the ground is different from the reference. Now you could use the ground also as a reference, but usually it produces more noise. So we have one ground electrode, which is essentially a path for all the induced charges to go, which is produced by all this electromagnetic stuff around us. And then there's a reference electrode, which is the electrode to which all other electrodes are referenced, which means that the eventual potential is just whatever you are measuring at a particular location minus the reference voltage. Okay, does that make sense? That is why we have two different electrodes here. So what do you think is a problem in choosing a single electrode here as the reference?
Neural signatures in other parts of brain. Those can have uh, different landscape than the central reference electrode. And
Yeah, so the problem is that's right. So suppose when you present a stimulus, there is some activity in this part of the brain. Okay, so suppose there is some activity in this part of the brain and everyone else is silent. So suppose a hypothetical case where the stimulus produced a response here, but no response in the other electrodes. But because I'm subtracting this from that and looking at that, I will actually see a response in every electrode. And it's going to be exactly the same response because the true potential here is nothing, but this one has some activity. So this minus that is just going to be then the mirror image of this that you will see on each electrode. And that's not good, right? Because you will attribute it to some activity that you are looking here, but then it's going to be reflected on all electrodes because it's actually the activity that the reference electrode has put in. Okay, so that is why when we do these recordings, you want to have the reference electrode where you don't expect too much activity. Some people, for example, use the ear lobes as the reference. So they will have the reference electrode on the two ear lobes that they will then connect and use that as the reference. Some people put the reference on the forehead. People have even tried putting the reference at other parts of the body. So the problem is that if when the reference electrode becomes too far away from where the activity is, then there is more noise, because if there is some noise being captured, this is again the line noise, not the true signal. You want the reference electrode to also have more or less the same noise so that they get subtracted out. So the farther it goes, if you put it on the leg, for example, it's going to catch different noise than what you have here. And then the subtraction won't eliminate the noise as much. So that's why you want the reference to be as close as possible. But at the same time, if you put it too close to the electrode, then essentially you are cancelling out physiological signal as well, because whatever this guy is catching, suppose if this was the reference, the neighboring electrode, then it would more or less catch the same thing and then everything will get subtracted out. So there's actually no unique good answer to what the reference electrode should be. If it's too far, then the signals that you get, the output is very noisy. If it's too close, then you get nothing. So different people have used different strategies, and here are a few common standard ones. One strategy is that you take the average of all the signals. So you just take all these 64 electrodes and take the average of that and use that as the reference. This is called average reference. It's a very popular strategy. Another strategy is what is called bipolar reference, where each electrode is simply referenced to its neighbor. Here what you mean is that when you look at this electrode here, for example, you will reference it with respect to this one, which means you are going to look at OZ minus O2 and think of it as a virtual electrode sitting here in the middle. Now you may say, why on earth should I do that? Because you will cancel all this, physiological signal which both of these electrodes are picking, and that's true. But anything that is very localized to this electrode but not to its neighbor will get picked up. So bipolar referencing actually picks up very localized activity. Okay, so from these set of electrodes, you can actually think of a virtual array of bipolar electrodes that correspond to, this minus this would be a virtual electrode here, this minus that would be a virtual electrode here, this minus this would be something like that. So you can have a lot of virtual electrodes sitting between the actual electrodes, and the value here is just this minus that, that minus that, and so on, and actually have a bipolar distribution computed this way. And this is only going to pick up very, very localized activity, which is not seen in the neighboring electrode. So gamma response, for example, is actually very well picked up if you do a bipolar reference. And we'll see that in a bit. So these are two popular schemes. In fact, average reference is the most popular one. Bipolar is useful for looking at gamma oscillations, for example. And then there is another reference scheme called source current density, or CSD, where you average the neighboring 4 electrodes and reference to that. So your signal, for example, for this one would be C3 minus the average of these 4. Okay, and we will discuss shortly why this is a popular strategy, current source density. So in fact, we'll develop the math why this is done. The code that I have given you allows you to look at different reference schemes. Here in display data, you could change this to average bipolar or CSD. So let me do that. Let me do bipolar here. I will do this on that. Okay. Now it's giving me an error because, oh yeah. Yeah. So there's another software called EEG Lab, which I'm using right now for something else. And that also part has a field trip partial. So there is some conflict between the two. So when you are running this, make sure both are not installed at the same time. This leads to some problem. Okay, so here I have field trip installed, but I also have EEG Lab, and that's what it doesn't like. So I have to uninstall EEG Lab, and let's see if that fixes it. No. Okay, I will see what is wrong here and try to fix that. But it used to work. Let's check average referencing. Okay, I'm running into some problems here. I think because they were overloaded, and the defaults are not set properly. So if I reinstall it, it should work. If it doesn't work, let me know. We will check what is the problem. But I actually have saved the data, so you can see how the topo plots of gamma look like for different referencing schemes. This is the original reference which we saw, and the average reference basically doesn't do much. But if you do bipolar referencing, you see that you have now finer resolution. So you can see more pockets here and so on. And that is because you have referenced each signal to its immediate neighbor. And look at the dot distribution here, these black dots. This is different from these ones because these dots correspond to the sensor space, and here it's these virtual sensors, which is the subtraction of the average of this and this. So when you do this minus that, you put something in the middle. Okay, so the bipolar referencing gives much better resolution. Now turns out referencing has a lot of other issues as well. We wrote a paper also on this. It changes the shape of the power spectral density. So if you look at the signal using single or average or bipolar or CSD, you can see that the spectral density, the shape of the spectral density, it sort of flattens out as you go from single average to single to average to bipolar and CSD. It reduces. Why does it reduce? The simple idea here is that um, so there's a lot of low power at lower energy, right? So the signal has this power law shape, but the lower frequencies, the low frequency sinusoids are also more coherent. You may have remembered that if you do field coherence, then you see higher coherence at lower frequencies, which means that the lower frequencies are more similar to each other across the scalp. And therefore, if you subtract it, there's a larger reduction of the signal as compared to higher frequencies, causing this slope to fall. Okay, does that make sense? The lower frequencies are more coherent. We have seen this in the LFP data. It is true for all types of signals, including EEG data. So when you are referencing, you are subtracting out a similar looking signal at lower frequencies, but not so much, a lesser similar signal at higher frequencies. So the reduction is more at lower frequencies, causing the slope to fall down. Okay. Turns out this is a more complicated finding, that it changes the phase coherence as well. But we won't talk about this. If you're interested in figuring this out, you're welcome to read this, or you can try out in the data that you have, that the phase coherence actually shifts from 0 to 90 degrees when you do the average reference. And the reason is quite non-intuitive, and maybe I'll give this as a homework assignment for you guys. But it's a very sort of interesting finding, that not only uh, the the PSD slope changes, which is straightforward to understand, but the phase difference also changes when you do an average reference. Okay, but I will not get into this in too much detail because it's very nuanced and sort of takes a while to understand. Yeah, give this as a homework assignment, and if you read this paper, you'll be able to figure it out. Okay, so instead I'm going to talk about CSD, which will lead us to this question of forward and backward inverse source localization. So this is now we are going to talk about a bit of physics. Okay, so the physicists here will like this part a lot. Um, so let's get started on this. So here's the idea. I have given you the voltage distribution on the scalp. Okay, so I've given you 64 time series data. Where's the raw? Yeah, but they are all voltage traces, voltage as a function of time. Why is this voltage getting generated? Well, this voltage is getting generated because when I present a stimulus, there are these miniature current sinks and current sources that are produced by the neurons. These are producing little dipoles, and we are picking out the contribution of those dipoles here as a voltage. Now, from voltage, I want to get back to the charge distribution. Okay, so given a voltage, can I find out the charge that led to this voltage? Going from voltage to charge. How do I do that? So here's the answer. This is not something that you can intuitively figure out. This is where you need to get to, electromagnetism. The procedure is as follows. From the electric potential given here as VE, which is a scalar quantity, you can compute what is called an electric field. And the electric field is just what is called the gradient of the voltage with a negative sign. Okay, so this inverted triangle is called a gradient, and the gradient of f is defined as this: is the change in f along the x direction multiplied by the I vector, which is a unit vector pointing in the x direction, plus what you see in the y direction and the z direction. So the way voltage changes in the xyz plane allows us to estimate the electric field, which is now a vector. Okay. So if I give you the voltage at one location, you can't compute the electric field. But if I give you the voltage at many locations, so if I give you V as a function of x, y, and z, I will be able to estimate the electric field by applying this formula. Okay, so note that you need to know V as a function of x, y, z for you to compute this. Okay, so you say maybe you guys remember old high school physics where you would have a plus here and a minus here. These are the charges, and then you would have these electric field lines, or maybe it goes from positive to negative, I think, like this. Does anyone remember this? Electric field lines. So this is how you would compute the electric field lines for given a charge distribution. So given a charge distribution, suppose you knew the voltage at all in this entire space at any point x and y, x, y, and z. So it's a scalar value at each location. You could use this formula and get to the electric field. Now the question is, hey, why do you care about the electric field? I wanted to know about the charge distribution. And the reason you want to know about the electric field is because there's a way for us to go from the electric field to the charge distribution. So here's the charge distribution that I'm interested in. This electric field is the intermediate step, and there's the voltage that I recalled from the scalp. What's the distribution relationship between charge distribution and electric field? It's given by this formula. This is what is called the divergence, this rho dot E, and it's just the dot product of this operator rho, which is given here. So you can think of this being an operator, I plus del y j. So this is the del operator. If you simply apply to f, you get this, and this is the gradient. And if you take a dot product with E, so taking the dot product essentially gives you back a scalar, but it's the change in V1 plus the change in V2 plus the change in V3, where V1, V2, V3 are the voltages in the X, Y, and Z direction, and you sum them up. This gives the divergence, and that is what is equal to the charge normalized by what is called this electric constant, or you know, denoted by epsilon. So if you have a charge sitting somewhere at the center, your electric field lines are just spreading out like this. And then the divergence of this quantity, because they are both all going away from this, it will be a positive value here. If there's a negative charge here, electric field lines will all be going negative, going inside. So the rate in change is always negative. So this is going to be a negative number here. And if the electric field lines are just passing like this, the divergence is 0. Okay, so the way these electric field lines are spreading out tells you whether there's a charge sitting here. Going back here, the electric field lines will all be going out from this. And then if you compute the divergence at this point, you will find out that there's a charge sitting here. Likewise, you will find a charge sitting here. And if you compute the divergence at these points, you will get 0, because there's no charge sitting here. So putting all of this together, the way to compute the charge distribution, which is what we are interested in, because I want to know where the charges are that led to the potential here. The idea is that I just take the gradient of the divergence of the gradient, which is basically nothing but the double derivative of the voltage. This is what is called a Laplacian operator, and that's all you need to do to estimate the charge from the voltage. Remember, I recalled the voltage here. I want to find out what the charge is. All I need to do is compute the Laplacian, which is the double derivative operator here, and then you add it up to get this. This is a scalar, and so is this. There's no vector here. But to get to this formula, I needed to get the electric field. Okay, so we will continue with this in the next class. I have run out of time. This was a little bit involved, but you know, this is the key concept that we will keep using as we make it more and more complicated. Okay, so in the next class, we will deal with this in more detail, and then talk about algorithms that estimate this, you know, charge distribution, and gives you what is called the source map from the scalp map. Okay, so we are done. I will stop recording now, and as usual, if there are any questions, please ask.